So one thing that we began introducing in this block course is just very short lectures about research actually going on in our labs, because we realize that what we do in the block course, of course, is somewhat related to what we do, but it's a nice opportunity for you as students to get to know what kinds of things we do in the lab, what kind of approaches we use, and the questions we're asking. So I'm going to give a brief research vignette today, and Peter is going to integrate his research into his lecture tomorrow. So what I just wanted to do then is give an introduction to the research in the lab. So I'm just going to start this. Give me a moment. Okay. One more second. Just getting all these windows sorted out. All right. Great. Okay. So, research in my lab is focused on this fascinating question of stem cells in the adult brain, which are turning out to be a really intriguing source of new cells and may represent a very important form of brain plasticity in the adult uh, animal. So, oh. Okay, as you know, stem cells are found in many different organs, and they live in highly complex niches. And these niches are beginning to be identified in many organs. And as I mentioned yesterday, stem cells are often found in these dormant or quiescent states and activated or actively dividing states. But very little is known about what triggers the activation of a quiescent stem cell, and what are the lineage relationships between these. Once it's activated, does it go back to quiescence, or does it continue down the lineage trajectory? So we're interested in these questions in the adult brain. Now, in the brain, you have neurons. You can see here a beautiful picture from Ramon Cajal highlighting the complexity of some of these different types of neurons. But you also have glial cells, and glia are often forgotten. Um, you have astrocytes, which have this star shape. That's where they get their name, and they're very important for as support cells in the brain. They participate in ensuring that synaptic efficacy is correct. They participate in forming the blood-brain barrier, which stops signals in the blood from entering the brain. And they are also important for maintaining neuronal homeostasis. The other main class of glial cell are oligodendrocytes, and these cells form myelin sheaths along neurons, the axons of neurons, and this allows the neurons to fire very rapid action potentials. So most neurons are born in embryogenesis and stop being born around birth, but glia can continue to be born throughout life. What people often forget when they look at the brain is that actually there are other cell types there as well. So this is a picture showing the blood vessels in the human brain. And you can see they form this amazing dense network of cells, so they occupy a lot of space in the brain. The other important fluid, which is often forgotten, is called the cerebral spinal fluid. So the CSF is found in the ventricles, shown here, in the brain. And it's important, it is found in the ventricles, but also around the surface of the brain. And it's very important for protecting the brain against shocks and also for kind of filtering out toxic substances. But we now realize that it's probably more important than just having these support roles. So, although neurons were thought to be born only during embryonic development, work over the last 2 decades has shown that actually new neurons continue to be born throughout adulthood in the brains of many different species. So, this was actually first shown in adult songbirds, and it was shown that they add new projection neurons that are important for song learning. And then mammals, it's been shown, this is actually the very first studies were done in mammals. And then since then, it's been found in fish and in lizards and primates. And it's still controversial as to how much neurogenesis is ongoing in humans, but there are clearly stem cells there. So what's interesting is that depending on the species, the numbers of neurons that are generated and also their destinations are quite different, suggesting that these new neurons may be important for brain function. So in our lab, we're focused in the adult mouse brain, and this is a top-down view of the mouse brain. And in particular, we look at one stem cell niche called the ventricular subventricular zone, which lies along the walls of the lateral ventricles. So it's outlined here in a blue shape. And the easiest way to visualize the whole stem cell niche is to just dissect it out as a whole mount preparation. And when you do that and stain it, you can see that the whole SVZ is traversed by this amazing network of newly generated neurons. So each one of these brown circles is a new neuron. So about 30,000 to 100,000 new neurons are born every day in this part of the brain, and they migrate most of the length of the brain to reach the olfactory bulb, which is important for the sense of smell. And in the olfactory bulb, these interneurons integrate into the circuitry, and they can be important for modulating the olfactory responses. So if we take a cross section through the SVZ, so across here, this is where the SVZ is located. So here are the ventricles, and the SVZ is found around the ventricles. Now, on this lateral wall, which is the most neurogenic site, you have active neurogenesis and many dividing cells. And on the septal wall, you have many fewer dividing cells. So if we zoom in here into this area, we can see the cell types that are found in the stem cell niche. So first you have these ependymal cells, which have long cilia, and these cilia then beat the cerebral spinal fluid through the ventricular system. In blue, you have the stem cells, which are found in quiescent and in active states, and they touch the cerebral spinal fluid, and they have a small primary cilium that they can use to actually sense signals in the CSF. On the other side, they touch blood vessels, and these stem cells express a marker called GFAP, which was classically thought to be a hallmark of astrocytes in the brain. So these neural stem cells, once they become activated, give rise to transit amplifying cells, shown here in green, which then divide to give rise to neuroblasts, which then migrate to the olfactory bulb where they turn into mature neurons. So many years ago, we found that the stem cells were actually a kind of astrocyte. And this was a bit of a surprise because neuronal and glial lineages were thought for a long time to be separate lineages. So this raises the really interesting question of whether glia all over the brain are stem cells, or whether only those found in these stem cell niches can act as stem cells. And likewise, what are the signals or the microenvironment that supports these? So one way to tackle this question is to be able to purify the stem cells directly from the brain. And so we can do this using a procedure called fluorescence activated cell sorting. And so here we see a stem cell contacting the blood vessel. It has a very polarized morphology. And over the years, we've now identified a combination of surface markers that can be labeled the different populations of cells. So one difficulty in the field has been actually to isolate the quiescent stem cells because there weren't markers for them. So we now know that they express a surface marker CD133 and GFAP, and when they become activated, they begin to express EGF receptor. And then as you go down the lineage, these other markers are turned off and the cells then become neuroblasts. So if we just combine all these markers, we can actually purify very nicely cells at each stage of the stem cell lineage. So what we use are mice in which the GFAP promoter drives green fluorescent protein. So you can gate on these GFP positive cells and then ask which ones express EGF or bind EGF receptor, and which ones express CD133. And so here you can see the quiescent neural stem cells express low EGF and are negative for EGF and are high for CD133, and the activated neural stem cells express EGF receptor and CD133. So we can purify then these populations of cells and test their properties in vitro. We can do culture experiments, we can do transplantation experiments, and we can analyze the transcriptome of these cells to understand the gene regulatory pathways. So this can be done by single cell profiling or RNA sequencing of purified populations. So what this highlighted then, when we purified the quiescent stem cells, is that actually the quiescent stem cells are really actively maintained in this state. So they express many genes involved in cell-cell interactions and also adhesion to the niche. And they have quite distinct metabolic pathways, such as lipid metabolism, active in them than are present in the activated neural stem cells. And very interesting, they're highly enriched in G protein coupled receptor signaling, suggesting that they may be sensing many signals in the environment. So from our studies, we realized that the stem cells, even though we can isolate these quiescent stem cells, they're still a very heterogeneous population. So we can see different layers of heterogeneity in the SVC. First, you can see that the stem cells have different shapes, so their morphology is quite distinct. We can see that they have differ in their proliferation state. So some are quiescent and some are activated. And a really open question in the field is still whether it's the same population of stem cells that are recruited under injury or if you have a separate reserve pool of stem cells. And then finally, in the context of the SVZ niche, a very interesting finding is that the stem cells themselves have a regional identity. So depending on where they're located in the SVZ, they actually give rise to different types of neurons. So for example, this region here will give rise to superficial granule neurons, whereas this region will give rise to deep granule neurons. And other domains will give rise to different subtypes of periglomerular neurons. So what's kind of remarkable about this system is that many of these stem cells that are quiescent actually enter quiescence very early on during embryonic development, and then they maintain this state throughout adulthood until they become activated. So this raises the question of how are these cells maintained in this state, and then what are the signals that actually stimulate them to become activated? So this is a big focus in the lab. What maintains and regulates stem cell quiescence and activation? And this is due to a balance between the intrinsic state of the cell. So what are the repertoire of receptors does it express? What are the gene regulatory networks inside the cell that maintain the quiescent state? And then on the other side, what are the extrinsic signals or the niche signals that come and stimulate these stem cells or maintain them in the quiescent state? So I'm just going to highlight a few recent sort of directions that we're going in now to show you how the system is becoming incredibly interesting and complex. So first, if we think about the intrinsic properties of the cells. So what we recently found is that platelet-derived growth factor receptor beta is a novel regulator of adult neural stem cell quiescence. So you can see here in green, in a whole map preparation, the stem cells touch the ventricle at the center of pinwheel structures formed by the ependymal cells. So the stem cells express PDGF receptor beta. Here we can see their beautiful radial shape by expressing a reporter protein, M-cherry, under the PDGF receptor beta promoter. So these cells are really, the quiescent stem cells express PDGF receptor beta. The rest of the lineage is negative. So what can we do if we, so what happens if we delete PDGFR beta selectively in the stem cells? What happens to their behavior? So we can do this using genetic approaches. So you'll hear a lot about this tomorrow, but essentially we can use the human GFAP promoter driving the expression of a inducible Cre recombinase. So when you give tamoxifen to the mice, GFAP positive cells will activate Cre recombinase, and this will then bind to flock sites and delete the intervening sequence. So in this case, we have PDGF receptor beta and also a stop cassette. So when Cre is active, you lose the binding domain for PDGF receptor beta, and we have expression of tomato protein. So we can follow the cells that have been recombined. So after tamoxifen injection for a very brief period, 3 days, we then see that there's actually a really big increase in the number of activated neural stem cells. So the stem cells are being released from quiescence. So here on the right, you can see 2 dividing stem cells, and in a control mouse, you have 1 So this also results in more transit amplifying cells and neurons that migrate to the olfactory bulb. So what was interesting about this model is that it actually allowed us to begin to ask, are there other quiescent domains in the mouse that are normally not dividing, but that we can now visualize because we're releasing the stem cells from quiescence? When I briefly mentioned before that this side, the septal wall, is normally largely quiescent under normal conditions. So can we use these mutant mice, these PDGFR beta deleted mice, as a tool to identify new domains in vivo? So again, in a cross section, here we have the lateral wall and the septal wall. And what's been known for many years, as I showed you before, is the lateral wall gives rise to different types of neurons that migrate to the olfactory bulb. And then there's a small domain up here in orange, which gives rise to oligodendrocytes. What's been very unclear is whether the SVZ generates astrocytes, and if so, where they come from. So using this mice, we were actually able to find that there were several gliogenic domains in the SVZ that normally are quiescent under normal conditions. One of them, or largely quiescent, one of them here gives rise to astrocytes. And then we identified several additional domains that give rise to oligodendrocytes, one up here, and one very interesting one that's actually inside the brain ventricles. And these cells here, instead of being in the tissue, are sitting on the surface of the wall of the ventricle. And you can see them here, these beautiful green spider-like cells. They're very branchy, and they're literally bathed by the CSF. So these cells, it turns out, are actually touching axons, which are known to come from long distance brain areas in the brain to this part of the brain. And they contact these axons and wrap around them. So we don't know what they're doing, but it's a really interesting new cell type in the brain that seems to be perhaps sensing signals in the cerebral spinal fluid and interacting with information from other brain areas. So in addition to new neurons then, it's clear that the adult brain can generate new glia. And this raises this very interesting question of whether glia might also be contributing to brain plasticity. So this is something we'd like to follow up and understand in the future. So now I'm going to turn to the other side, and that's the niche. And as I've already explained, the niche is quite complex. Here in a whole mount, we have the stem cells contacting the ventricle at the center of the pinwheels formed by ependymal cells. You can see the stem cells have a single primary cilium, shown here with a single dot. And they have a very polarized morphology. So on this side, they're touching the CSF. And on this side, the basal side, they're contacting blood vessels. And then throughout the SVZ, you have a very specialized blood vessel network, which is quite flat, so it's a planar structure. And you can see here in green, the dividing cells right next to the blood vessels. Now, in between the CSF and the ependymal cells and the blood vessels, there are many other cells as well. So there's extensive local cell-cell interaction between stem cells, lineage cells, and other cell types in the brain, including extracellular matrix. So what we found actually is that the vascular niche is quite interesting. So in addition to secreting diffusible signals that can regulate the stem cells, the blood vessels in this part of the brain have unique features. So normally in the brain, you have astrocytes completely ensheathing the blood vessels, and this is part of the blood-brain barrier. So what we found is that actually the stem cells and their transit amplifying cells directly contact the blood vessels at special sites that are missing these astrocyte end feet. So they're uniquely poised to get signals directly from the blood vessel surfaces. We also found that factors in the blood, these small molecular weight factors, are able selectively to enter this part of the brain, suggesting that there's this systemic factors that can regulate neurogenesis. Now, on the other side, you see here the stem cells contact the CSF. Let's just step back a moment and look at a whole mouse brain here. And here we have the ventricles where the stem cell niche is found, the lateral ventricles, and the CSF flows through to the third ventricle and the fourth ventricle. So they're continuously bathed by the the stem cells are continuously bathed by the CSF. And the CSF itself is made by a structure that's floating inside the brain ventricles called the choroid plexus. You can see here it has this fan-like shape and it's highly vascularized. And what we found is actually the choroid plexus itself secretes many factors that are important to regulate the stem cells, but also their progeny. And very interestingly, the factors that the choroid plexus makes change in different states. So, for example, in an aged mouse, the factors that are secreted by the choroid plexus actually inhibit the division of young stem cells, but the factors from a young choroid plexus can stimulate the division of old stem cells. So, we have the idea then that the choroid plexus, which is at sort of sensing signals from the body because it's receiving blood, um, integrates these signals and then dynamically changes its secretome to then affect the stem cells and their niche. So, together, these really highlight the importance of long range signals, not just cell cell interactions and very local interactions. The big question that's been open in the field is really what activates quiescent neural stem cells to be turned on. So, to understand this, we FACS purified the quiescent stem cells and did a small molecule screen to try and identify compounds that might turn the stem cells on. And so this is just a simple readout. You can purify your quiescent neural stem cells and use a marker of cell division, shown here in red, called MCM2, and another marker for activated neural stem cells called nestin. And so in a control, you'll have occasional quiescent stem cells becoming activated. So we looked for compounds that would increase the individual number of quiescent stem cells becoming activated, or which also form larger colonies. So the interesting finding that emerged from this that it was actually neurotransmitters that stimulated the quiescent neural stem cells to become activated. This was the major class of compounds that did this, suggesting that maybe neural activity could be modulating stem cells. So this was interesting, and we followed up in one candidate in particular called beta endorphin. And when we looked in vivo to see where the beta endorphin might be coming from in the stem cell niche, we found a very interesting regionalized pattern. So just to remind you, here we have the SVZ, and what's known is that different domains within the SVZ express different transcription factors because of their regional identity. So this domain in blue, which is ventral, expresses the transcription factor called NKX 2.1. And when we looked for where the beta endorphin fibers were, as shown here in white, we found that they were really targeting only this ventral domain that overlaps with the green NKX 2.1 domain. And dorsally, you didn't see any fibers. So we then asked, where are these fibers coming from? And it turns out that they're coming from a structure in the brain called the hypothalamus, which is very important in different states, such as feeding and sleep behavior and mating behavior and other sort of natural physiological states. So one particular population of neurons in the hypothalamus, called POMC neurons, make beta endorphin. And you can see them labeled here. They're very far away from the brain ventricles. So we were able to selectively target these POMC neurons and then manipulate their activity and ask what happens to the stem cells in the SVZ. And really remarkably, we found that this population of POMC neurons sends long-range fibers that target this ventral NKX 2.1 positive neural stem cell domain and specifically regulates the division of this pool of stem cells. And this then stimulates the production of one subtype of granule neurons in the olfactory bulb. So what was really interesting then is that not just by manipulating the circuit could we trigger stem cell division, but just by sort of using a natural physiological state of hunger and satiety, so feeding or fasting, we were also able to stimulate and regulate the division of this NKX 2.1 positive neural stem cell domain. So this is leading us to a new sort of idea of understanding why there might be different pools of stem cells, that they may be important for different states, and that in fact, long-range neural circuits from other brain areas may be controlling the division of some of these pools of stem cells. So we can come back to our earlier model and see that at least some populations of stem cells may be quiescent and then turned on when they're needed. So we're very interested then in understanding this on-demand recruitment of stem cells in the adult brain. So we found one domain that's regulated by feeding and hunger and satiety. And now we know there are several new gliogenic domains that are normally quiescent under normal conditions. And so we would like to uncover what are the other states that regulate adult neural stem cell pools. Are these different between female mice and male mice? And what are the function eventually of these new neurons in these different states? Do they contribute to a kind of glial plasticity? And what regulates the recruitment of these different pools of stem cells? So, in an ongoing project in the lab, we found actually that pregnancy stimulates other domains to be turned on. And this raises really interesting questions about the function of these cells as well. So then in the end, what we'd like to understand is how neural circuits might regulate these pools of stem cells. Perhaps some come from the hypothalamus, some might come from other brain regions and impinge on these different stem cell niches. And to do this, we're trying to develop methods to visualize whole brain imaging. We'd also like to understand how, for instance, these new cell types we found, these interventricular glial progenitors, are regulated by the choroid plexus, and whether the choroid plexus might be dynamically changing in different states. So we're profiling the choroid plexus in young mice and old mice and males and females in other states and trying to understand how its secretome might be affecting these cells. And we can see already here, just simply RNA-seq analysis of a male and a female choroid plexus shows that there are quite a lot of genes that are differentially regulated between them. So finally, what we want to understand then is sort of integrating data sets on single cell analysis of the different populations of stem cells, the niche signals, and to understand how these all come together and regulate different pools of cells for on-demand neurogenesis and gliogenesis in male and female mice. And for this, we have a wonderful team of people in the Biot Centrum, many of whom you've met, some of whom you'll meet later in the block course. And formerly I was at Columbia. So thank you very much. Leave this here. And I'm happy to answer any questions. Does anybody have any questions? You need to raise your hand because I can't see things very well.
I have a question.
Yes?
Can you upload the slides on Adam?
Well, these, I wasn't going to because there are research slides.
Okay.
But that's why I didn't upload them because it's not important for your things.
Okay.
One thing, what is the relationship of meat to mind? So you mean food and mind? So, I mean, it's very interesting. So one of the odd things about this finding of the hunger and satiety and the regulation of specific populations of neurons is the time scale is kind of odd, right? So the stem cells divide and then they really form functional neurons only much later. So about 2 weeks to 4 weeks later. So the question is, why is there this delayed time frame and why might hunger and feeding affect the circuits? And so we don't have an answer to this. One idea is that perhaps in the wild, animals go through bouts of fasting and feeding. So this may be a way to kind of regulate stem cell populations to preserve them or to be more sensitive in states where you may need to acquire different sensory things. Oh, how does the brain make consciousness? That's it. Okay. So yes, that's a big question that perhaps other people would like to contribute to answering. So we don't know the answer, right? So it's an emergent property, but how does this happen? What do people think? Anybody have ideas? Okay. So one thing I realized, or that I and all of the faculty, is that one of the difficulties with this virtual online experience is that the students don't have as much opportunity to interact in real life with the tutors and also the faculty. And normally we'd be in the labs and we'd bump into each other and be able to talk with you about different things, be it your experiments or research. So we were wondering if it might be helpful to set up some kind of small forum or set a time where if people are interested in talking with us, we're happy to interact with you. Would that be something that's of interest to people? We could set a time, perhaps a couple of times, and you can we can open a Zoom link and people can come in and talk to us. Do people like this idea? Yes? Okay. Lots of thumbs up. Okay. So I'll talk with Peter and we can see if we can find some times when you don't have lectures or exercises that are being tutored, or you would have the opportunity to ask more questions. The other thing is that, and we'll come back to you about this, is of course this is a new experience for us and for everybody, and so we're very eager to get your feedback on what works, what doesn't work. Are we, what should we change in the future? Hopefully, we can always go back to doing practical experiments, but maybe there are some facets that we can integrate into future block courses. So we'll come back to you at the end of of our section and ask you, but please keep giving us feedback about ideas you may have. All right. So do you have any other thoughts or questions? No? Okay. So now today you're going to be going